Hello everyone. I'm Caroline Uhler, a faculty member in the Department of Electrical Engineering and Computer Science, as well as the Institute for Data Systems and Society at MIT. And I'm also a core institute member at the Broad, where I co-lead the Eric and Wendy Schmidt Center at the intersection of the biomedical sciences and machine learning. And it's really my pleasure to welcome everyone to this online symposium on machine learning-based biological, experimental, and therapeutic design, which is organized and sponsored by Immunai. Now, why are we having this workshop? Well, unlike many other fields in the biomedical sciences, it is really possible to perform these large-scale perturbation screens, which are critical in order to get at the underlying gene regulatory mechanisms. In particular, with the recent capabilities, we have access to large-scale genetic perturbations with which we can knock out or overexpress particular genes or even combinations of genes. And we can perform really also large-scale chemical perturbations and observe how cells respond to different drugs. But, one of the challenges, and of course also opportunities, is that the space of possible perturbations is so huge. And so we want to understand what genetic perturbations do to any gene and even any subset of genes. And similarly, the space of molecules is huge, right? We also care about the effect of combinations of drugs, for example. And, you don't only have this huge space of possible perturbations, but also perturbations can have very different effects in different disease contexts or in different cell types. So this workshop is about, how to deal with this combinatorial explosion of the space of perturbations and contexts. And we will discuss machine learning methods that we've been working on for this experimental design problem of biological and therapeutic interventions. And so I'm really excited to have two pioneers in this area here with me, and I'll introduce them afterwards when we're going into their talks, namely Regina Barzilay and Devavrat Shah. So the schedule for today is that I'll first discuss some of our recent work in the space of exploring and optimizing genetic perturbations. Then Devavrat will tell us more about his recent work on predicting the effect of unseen perturbation context pairs from observed perturbations and contexts. And then finally, Regina will tell us about her exciting work in the space of molecule design. And we'll have time for questions from the audience after each one of the talks. So let me get started with my talk, and I don't want to forget to mention that you should all see the Q&A feature on your screen. So please feel free to submit questions throughout the talks, and we'll then pick from these questions after each talk for a 5-minute Q&A. Okay, so let me try sharing. So let's do this, share the whole screen and pull up my slides. Okay. Let me go to the beginning of it. So let's just get started. So what I wanted to do is, again, just introduce a bit of the problem of what we've been looking at. So for us, it has mainly been thinking about, genetic interventions and this huge space of possible perturbations that you can do when you're performing genetic perturbations. As I said before, particularly, you also care about questions like combinations of genes. And then, of course, it will never be possible to perform all of these interventions. All combinatorial interventions of all, say, knockouts on 2 genes and 3 genes and 4 genes and 5 genes, given that we have 20,000 genes. So how do we go about this to optimally design interventions? And of course, optimal will really depend on, what you're optimizing for. And so I'll discuss this also a little bit. Okay, so the motivation is really coming from single cell biology. I mean, these amazing technologies where you can now really obtain these very high throughput observational and interventional single cell gene expression data sets. You can not only get this as in sequencing land, so single cell gene expression, but also in imaging land where you get to observe, like here on these slides, single cells, and you get to label them with all kinds of different fluorescently labeled marks and get to see what they are expressing. And together, we can observe these large-scale, with where one data set has, say, 100,000 cells. But in addition, we can, as I said, also perturb cells at in very high throughput. And these can be genetic perturbations or also small molecule and drug perturbations. And, the objectives when you're doing these perturbational screens can be from really trying to understand the underlying wiring diagram of the cell, these gene regulatory networks, all the way to trying to identify optimal perturbations to move cells between different states. And states here can be, the disease state, trying to move that to the healthy state, or, and then, of course, relating the different genetic perturbations, maybe to identify targets that you could then use for defining or developing molecules that could then bind to these targets and try to do drug discovery in this kind of way. So target identification for drug discovery. So if I go a little bit maybe more directly into one particular problem where we're thinking about cell state transitions, and something that we have been very excited about is cellular reprogramming. So here the states that you want to move cells between would be different cell types. But the questions are just the same when you're thinking about a cell state being a disease state and moving that to a healthy state. Here our states are different cell types, and we would like to identify perturbations that can move one cell type into another cell type. We know from these amazing experiments by Yamanaka that there are about 5 transcription factors were needed in order to move fibroblasts into into an induced pluripotent stem cell state, which then can be used to give rise to all of the different types of different cell types again. And so, if we're thinking about all of the different cell types that we have, well, we would like to be able to understand how to move any one cell type into any other cell type. And that's, of course, a huge optimization question, or a huge experimental design question of identifying, which ones are these transcription factors that you would have to add in order, or even drugs, right? You can also do chemical reprogramming. Which ones would be the molecules or these transcription factors that you would add in order to move from one cell type to another? This is a huge search space. If we're just thinking about transcription factors, we have about 2,000 of them. And now think of, if we really need, say, about 4 or 5 of these transcription factors, well, that's a search space of like 2,000, choose 5, where, and doing this for any pair of cell types is not going to be an endeavor where we can really reach it just experimentally. So we will need computational methods that will predict what these optimal interventions are. Okay, so trying to do this a little bit more mathematically. So this is a machine learning symposium where we're thinking through how should we even think about these problems from a mathematical perspective. A gene regulatory network is often represented by these directed graphs, right, where a gene then can turn on or can turn down another gene. So these would be these directed graphs. And, you can intervene on the system where you, for example, knock out a particular gene, or you overexpress a particular gene through interventions on this graph where you intervene on particular nodes, and these nodes would be called targets. So these gene regulatory networks, or in a computational way, these are represented by these graphs, which I'll define on the next slide, which are represented by structural equation models, which really tell you how the expression of a gene changes if I'm changing the upstream genes or the expression of the upstream genes. And again, I said at the beginning that probably, identifying the optimal perturbations depends on what you're optimizing, or what your objective function is, so what the underlying problem is. And I defined there 2 problems. One, you can say like, well, I would like to identify the perturbations that will tell me or provide me most information about this underlying gene regulatory network. So my goal is to really understand the underlying mechanisms, the underlying biological mechanisms in a particular disease context or in a particular cell type. So I want to learn this graph, this regulatory graph. And so what are the perturbations that will tell me the most about the underlying graph? Now, why do perturbations tell you something about the underlying graph? Well, a perturbation on a node, then, if I perturb here, for example, this gene 2, well, only the downstream genes actually change, right? So in this case, only, say, for example, gene 3 changes, gene 1 would not change because it is upstream. And so that this particular perturbation tells me something about which genes are actually downstream of me and which ones do not change, and therefore are, not comparable to me. Either they're upstream or they are, somewhere on a branch that is not has nothing to do with the gene that I am in. So these perturbations can tell you something about these directions of these edges. So that's why they provide you information, structural information about this gene regulatory network. So that can be one goal. The other goal can be to try that you have two different states, like say two different cell types. I had in the previous slide, I have fibroblasts and I have these induced pluripotent stem cells. And I would like to find the interventions so that, I can move this distribution here, which is a distribution over fibroblasts and gene expression land, to look like the distribution over induced pluripotent stem cells. So it's really identifying the optimal perturbations that move one distribution into another distribution. Now, one way of doing this is by going through AIM-1. So by, if I learn the full underlying gene regulatory networks in fibroblasts, well, then that will tell me what the effect is on intervening on any one of my genes. And that's one way to then be able to move this particular distribution over fibroblasts to look like a distribution over induced pluripotent stem cells. But that might not be the optimal way, right? If all you care about is actually to move this distribution, then maybe you don't need to learn the full underlying causal graph or the full underlying gene regulatory network. There might be more cost efficient or with less perturbations, I might be able to actually just do this particular task here. And so that's already a theoretical question, right? Of like, can we do this second task? If this is really our goal, well, then how different are the optimal perturbations that we get here as compared to when we just try to do perturbations that try to learn the most about the underlying gene regulatory network? So that's what I want to just briefly talk about here in this talk, also a bit mathematically, to really see how do we think about optimal perturbations and what can we say so far about, what are strategies for actually identifying these optimal perturbations. Okay, so this is the framework that we use when we're thinking about these directed graphs or these causal graphs and thinking about interventions. Is a framework that is known as these structural equation models. It was already introduced in the 1920s by actually a geneticist, Sewell Wright, who thought about heredity. And so, these directed graphs represent direct causal relationships. So think of these as genes, right? And here, for example, gene 4 is a function of genes of the direct parents. So in this case, gene 2 and gene 3, and some noise. Of course, in gene expression data, they are stochastic. That's why you always have these noise terms here. Now, this can be linear relationships in the simplest setting. So, gene 4's expression is some linear combination of gene 2's expression and gene 3's expression and some noise, but they can also be just general functions. And so everything will hold also for general functions. Now, how do we think about interventions? And so now here, this is a framework that was developed by Frederick Eberhardt of thinking of distinguishing between different types of interventions. So in particular, let's think of, say the most extreme interventions, which would be knockout interventions, where I'm just taking a gene and setting its expression to 0. That would be an intervention that's known as a hard intervention in the machine learning context. So I would take here a gene and just set its expression to 0. So that means that then, if I'm taking a gene and just knocking it out, well, then all of the upstream genes have no effect on me anymore because I'm just set to 0, right? So in some sense, I'm changing the graph structure. Now, when I'm knocked out, then there are no more incoming edges into me. So that's known as a hard intervention, just going in and setting a node to a particular value. Soft interventions is basically everything else, and in particular, it encompasses things like, overexpression experiments that you may want to think of, and that we will be thinking of in particular in the setting of reprogramming, where you're going in and, say, for example, here, X2 is just going to be set to some higher value. So you're adding, say, some transcription factor, so that X2 is up regulated. But then, of course, the expression of X1 still matters, right? If it's direct parents. So if X2's expression was already very high, now we're adding a little bit, then it's still high, but it's at a very high level because it already started off at a very high level. So the expression of X1 actually still matters.
Is on.
Was there a question? Okay, I heard someone quickly, so I'll just continue. So there is this general framework for causal inference from observational data, which already started in the 1970s. But now, of course, having access to perturbations is a whole new challenge and, leads to completely new kinds of questions. Okay, So with this, let me go into the particular question of, let's look at our two goals of trying to identify the perturbations that give us the most information about the underlying gene regulatory network, and then looking at perturbations that could actually move us from one state to the other. And I do want to just, give a little bit of mathematical intuition for what these optimal perturbations are and why this problem is maybe hard as well. And what are like different kinds of tools from machine learning that we can use in order to come up with such strategies for identifying the optimal perturbations. So let's do the following first. So, if we want to identify the interventions that give me the most information about this underlying directed graph, right, which would be this gene regulatory network, then first of all, what happens if you intervene? So if I intervene on a node, we already saw that, it tells us something about the nodes around it, right? It tells us that, this, because I'll see that X4 here, this gene changes, it tells me that X4 has to be downstream. Since, it's quite easy to learn undirected graphs, but the difficulty is always the direction. So say, here I get to see that X1 and X2, they are correlated, there is an undirected edge between them, but I will actually be able to infer that this edge is pointed towards X2, because if I perturb X2, then X1 doesn't change. So somehow if you perturb a node, then it's possible to direct all of the neighboring edges. So what the intuition then is that, there's quite a lot of theory on just perturbing single nodes in a network. But this already tells you that probably it's actually better to perturb more nodes at once, right? So for example, if I perturb here X2 and X3, well, then I will be able to identify the full graph, right? I will be able to identify all of the directions in this network. So that's quite interesting. And so since in genetics, it is possible nowadays to perform these combinatorial perturbations where you go out or in and actually knock out multiple genes at once, well, that intuition then tells us that that's what we should be doing, right, in order to obtain the most information about the underlying gene regulatory network. So, but that's then a problem, because now we're now not only searching over the best single node intervention, and that's already a large space because we have 20,000 genes, but now we're looking at, the best combination of perturbations that we should be performing. And there is also something else that is happening here, namely, and the intuition here will already be clear, and I have it just written out on the next slide, is that if you have two genes that are neighboring each other, let's just say we have two genes, X1 upregulates X2. Well, if you knock out both genes at the same time, then you get no information about the underlying graph. So it's bad for you to knock out genes that are neighboring in the network, because then you don't get any information about the relationship, about the causal relationship between them. So it is not the case that just strictly performing more interventions in the network is better in terms of learning something about the underlying gene regulatory network. So that's a little bit of a problem. So generally, perturbing multiple nodes in a network is good. It increases identifiability. However, it's not strictly increasing. So it's not the case that, I should just always intervene on all nodes and that will give me the most information, because then you get no information about the underlying network itself. Okay, so there is this difficulty, right? And it is important that we try to understand what are the optimal perturbations to perform. Okay, so how do we go about this? And so what we're using here as a mathematical concept is, and first of all, what are objective functions that you're trying to use in order to optimize, or in order to define what it even means to be a most informative intervention? So there are different kinds of objective functions. And here I'll just use a very intuitive one, which was defined in this paper by Goswami et al. In 2018, which is, say you come in with a whole lot of observational data. So then you have some prior knowledge of, what kinds of graphs are possible graphs. Even say some edges are undirected. You might have, some prior based on the data that you've already collected on that your true graph lies within this equivalence class of graphs. I'll just call this an equivalence class of graphs. It's just a collection of graphs. And now you want to identify which ones of these collection of graphs is actually the true underlying gene regulatory network. Okay? So I need to get a score, and I need to identify which intervention should I do in this collection of graphs so that I can really identify which ones of all of these is actually the right one. And one of the scores that has been introduced is to score interventions is that, if I intervene on this and this node, say, how many more edges can I orient in this graph? Okay, the more edges I orient, well, the more I can actually identify which one is actually the true underlying causal graph. So that would be our score for every intervention. An intervention can have multiple targets, is, how many additional edges can actually this particular intervention orient in this equivalence class of graphs that I have in front of me. So I would like to identify the interventions that can orient the most edges in this equivalence class of graphs. So this would be the objective function that we're trying to optimize over. And so here, in terms of algorithms that we'll get to, we'll have algorithms that can run in general, but in terms of actually proving something about when are we really getting the optimal intervention out, we cannot have any latent variables, so all of our nodes are observed. If we're running this on gene expression data, then of course it's quite nice that we can nowadays observe 20,000 genes, but of course there's still going to be some latents, and so in that setting we will not have any guarantees. We'll be doing this for hard interventions, so this is knockout interventions. And again, the guarantees will always be in the noiseless setting. So that means that we will have infinite amount of data, which of course is not the case, but you can run the algorithms, you just don't have the same guarantees. So what are the challenges? Well, the challenges are the ones that I already described before, that we're not only optimizing over single node interventions that you want to get, but actually we are trying to do, in every intervention, we can have multiple targets. So you have this huge combinatorial search space that you're trying to optimize over. And, even from a prior, you might actually be averaging here over a large class of graphs, because you might have a lot of uncertainty over what the class of graphs is that you're actually looking over at the beginning. So there are these two very computational kinds of questions that we're trying to get over. So here is like the main intuition from a mathematical context of what you can use in order to get around this problem. And it's, I think, quite interesting as a concept, a mathematical concept that has been used in many, many different kinds of applications, that you can also use this for finding optimal interventions. It's known as submodularity, so the diminishing returns property, which is very intuitive in the intervention setting. It just says that, if I already have a lot of interventional data, then getting an additional intervention doesn't provide you as much additional information as if I only had very little interventional data. It's diminishing returns, right? If I already have a whole lot of data of a type, then probably getting one more sample of that type doesn't give me as much information as getting one sample of that type when I have very little of that type of information. And so that's something that is intuitive in the interventional setting, and is something that we can use in order to come up with greedy algorithms for identifying what these optimal perturbations are. And the difficulty is that, as I mentioned before, submodularity here is actually not monotone. It's not the case just that intervening on more targets actually always gives you more information. But there are versions of submodularity that you can use in order to also get greedy algorithms, even in this setting when you don't have monotonicity, to get algorithms that tell you which interventions you should do. So which targets to select for every intervention, and then which interventions to select for every batch. And you can get, proofs of how well this does because of the submodularity property. So you can say that, the strategies that you're getting are close to optimal. So they're not provably optimal. They cannot be, because this problem is, NP hard, but they are close to being optimal, and you even know how far away they are from being optimal. Okay, so how do these kinds of things work? They do outperform. And what is interesting to see is maybe, not just comparing to some other random baselines, but maybe what is interesting to see is actually to compare it to something that is greedy, that uses submodularity, but that can only intervene on single nodes. Right. So the advantage here is that now we can actually build these perturbations that actually intervene on multiple nodes at the same time. And these actually really strictly do better than when you're allowing yourself only to intervene on single nodes in every intervention, in every experiment. So that's what you would hope to see, right? That's why we were building all of these tools, because nowadays in genetics we can perform these interventions where you're actually perturbing multiple nodes at the same time. So I'm looking at the time, and I see that the time is up, so I will stop here and maybe just jump all the way to the last slide to say that, these kinds of similar strategies we can also use for not just identifying the most information about the underlying gene regulatory network, so identifying perturbations that are optimal in terms of information content in the underlying gene regulatory network, but also, and let me just move to the last slide, but also for actually trying to move from one state to another. So you have, your only goal is not just to find the most information about the underlying cell type, say the underlying gene regulatory network, but actually all you want to do is take, a cell type A and move it to a cell type B. That doesn't in general require you to learn the full underlying gene regulatory network. And we actually show that if this is your only goal, that you can actually can do this with the number of experiments being just logarithmically in what you would need when you're actually trying to learn the full underlying causal gene regulatory network. So if this is your only goal of just directly, trying to move from one cell state to another cell state, then the optimal strategies look very different to the optimal strategies that we have introduced here. And you can actually do these kinds of transformations with much less interventions than what you would in order to learn the full underlying gene regulatory network. Of course, a whole lot of problems remain open. We do have, so these algorithms actually here now, we can run them on up to 1,000 nodes. 100 nodes is good. We have run them on up to 1,000 nodes, but 20,000 is not in reach. We have also, as I said here, this was for knockout experiments. We do have here also something for these for soft interventions, but very special types of soft interventions where all we're changing is just changing the mean. We have not looked at, interventions that also change the variance, which is of course the case when you're doing, say, overexpression experiments. And yes, we have strategies that can run for these large-scale problems, but we have no guarantees in the noisy setting. All of the guarantees, while you can run it in the noisy setting, all of the guarantees that we have theoretically always are in the noiseless setting. So when you would have an infinite amount of data, so a whole lot remains to be done, which is of course exciting from our side. With this, let me end and I move back so that I see everyone, and in particular, if there are questions, I will now stop sharing. So I think I stopped sharing. I see myself again, and we'll take any questions. So I see down there is a question. Whether there are any computational approaches for multi-target drug design. And so I think this is actually very, very interesting and will lead over to Regina's talk. And so maybe that will be a question for more Regina's talk. So here it is about the identification of targets, right? And what these targets actually do inside the cell. And then afterwards, you would either have drugs that are, it's already hard to go from targets to drugs. But then here you would look at either drugs for the particular targets. And, it is a very interesting question of asking for drugs that directly target the multiple targets themselves. There, I think what is really exciting is that, and we have done a little bit of this in terms of target identification, is also to look at how the DNA is packed. Because often, when you actually find these multiple targets that are related to the same kind of cell state transition, etc., it's also because you have, say, a transcription factor that, often like the targets of this particular transcription factor are already close by inside how the genome is folded, because the packing of the DNA is optimized for particular functions that every cell type has to take. And therefore, you see these hubs of like genes that are already close by because they are being co-regulated. And so it would be actually very interesting to use this type of, spatial organization also to think through how to develop new drugs that could directly target different kinds of genes at the same time. Any other questions that I can take? Okay, and otherwise, I think it's actually the perfect time for me to introduce our next speaker. So and I hope Devavrat is here.
Caroline.
Maybe Devavrat, you can just briefly say that you are here.
I,
You're
yes,
here. Okay, perfect.
I'm
Then
here.
I
Can
can
you hear me?
introduce you. So, Devavrat Shah is the Andrew and Erna Viterbi Professor of Electrical Engineering and Computer Science at MIT, where he also directs the Deshpande Center for Technology Innovation. Devavrat has made many seminal contributions to statistical inference and stochastic networks, and he has also very successfully co-founded startups such as Select, which is now a part of Nike, which was the origin for, at least from what I know, which was the origin of some of his work in the space of interventions and causality. And Devavrat, maybe you will tell us more about that in your talk. So with this, I'll
Well,
hand over to you.
thank you, Caroline, for kind introduction. Thank you, everyone, for joining. What I'll do next is quickly share my slides and get us started. I hope it's
We see it,
visible,
yeah.
and I will make sure that I fully present it. Okay, and it's still visible, right?
Yeah, but not in presenter mode.
Oh,
So we had, I think, the problem before. So maybe if you go back out and you share your full, you don't just share the slides, but
how
you
about,
share your full screen.
is this still in presentable mode thing?
Yeah, it's still in not the right mode.
I see. Got it. Okay, so what should I do then? I should just share
Maybe
the
you
whole
stop
screen.
sharing. Yeah,
Yeah.
share full screen. Yeah.
Okay. Let's say share entire screen. Okay. And then I go here and then I do the full screen thing here.
Yeah.
Awesome. Now it's fine, right?
Thank you. This is
Yep.
perfect. Yes.
Perfect. Okay, great. So I have a very simple agenda in next 20 minutes. I want to, hello, can you still hear me? Yeah, okay. I have a very simple agenda for next 20 minutes. I want to talk to you about one case study in the context of a phase 3 clinical trial data from Taurex Therapeutics for Alzheimer's disease. And what we want to do is that we want to take the population level data available in this phase 2 clinical trial and try to see what sorts of personalized predictions we can make using a causal inference method called synthetic intervention. This is based on joint work with Anish Agrawal and Dennis Chen, who were former graduates from our department, and now both of them are at Berkeley doing their postdocs. Vishal Mishra, who is a faculty member at Columbia University, and Beyond Shelter, Helen Shields, and Claude Bishik are at Taurex Therapeutics. All right, so what I want to do is I want to quickly describe the question, the data set, the method. And my hope is that in 20 minutes or so, I'll be able to get through all of that. Okay, so here was the setting. Setting is a clinical trial that spanned over 18 months. 1,162 patients were enrolled. There were 4 different types of treatments were given. There was a randomized assignment, but not uniform, and hence you see different proportionality here. Out of these assignees, these were the individuals who complied, and these many individuals dropped out. So as you can see, roughly a quarter of the individuals dropped out and rest complied. Okay, and for each of the individual, over the period of 18 months at a regular interval, there were 5 clinical measurements were obtained. And as you know, if you would know in the context of Alzheimer's, one of the clinical measurement of interest, what's called also ADAS-COG score. And it's higher the score, worse it is, so you want to keep it small. So really, you don't want it to increase. And for each of these different 4 drugs, on average, on these populations, these were the outcomes. And as you can see, for different things, different outcomes were there. In particular, drug 2 was of really interest to them. There was a negative change on average. However, standard deviation is very high. So good outcome, but not conclusive because it's not statistically significant. And then in addition, of course, there is a 25% of patients that dropped out. So there is a biased because of that. So while, as we all know, randomized controlled trials are try to avoid the biases, but there is bias that always comes up because of the dropouts. So our question was that, can we on one hand understand that whether if the mean is good, maybe there are subpopulations that might be more effective than others. And second is, can we correct for biases induced by dropout? So those were the two basic questions of interest. Okay. Just to give a little bit more color on the data, for each patient, in addition to these 5 clinical measurements, we had 4 covariates that were measured before the treatments were assigned. There were 2 clinical measurements coming in, age and gender. So really, it's very simple sets of covariates. And based on these covariates, for example, one could do natural clustering, and it turns out there's a nice clustering that happens into 6 clusters. Here's just a PCA visualization of that. So putting it another way, as we started the study, we said, let's assume that there are 6 these clusters of the patients that are there, the 6 patient types, okay, based on these pre-treatment covariates, not looking at any post-treatment data. And each one of them potentially is given 4, or each of these patient group is given 4 treatments. And we want to understand which of these is best for each of the patient type. And high variance is a proxy of suggesting that there is some heterogeneity that's happening here. Putting visually, now we have effectively the situation like this. We've got 4 drugs of interest, 6 patient type, and I would like to know answer to each of these boxes. That is, what would be the treatment effect for patient group type 1 under treatment 0, treatment 1, and so on. And those are these boxes present. So really those 24 causal estimates are of my interest. And as I'm doing that, I would also like to make sure that I'm correcting for the bias introduced by dropouts. Okay. So with that, at least at the highest level, the question is set up. Now, what we'll do is the following. First, since in this case, turns out because of enough coverage of the data naturally, there is a ground truth we have, and hence it's a nice cross-validation setting for us. And question is sort of, can we try to fill in some of these cells without having access to them? Putting it other way, if we wanted to actually fill in all of these cells for each treatment, patient type, that is 6 times 4, 24 possible things, we would like to run an experiment. And as you know, experiments are expensive in this setting. Question is that, can we actually predict outcome of all of these experiments, but without conducting all 24 experiments? Maybe just half of them. Maybe all I want to do is 12 experiments. And can we still do that? And as we are doing that in the process, we also want to correct the bias induced by dropouts. Okay. So in summary, here's a setting that we will consider. So imagine that to the method, to the algorithm, to the problem solver, we will give data of the following type. For treatment 0, for all the patient type, we have the data available for patient groups here. That means these 6 experiments are done. For treatment 1, only patient type 1 and 2 patients are enrolled. That means only these 2 experiments are done, and these missing values in the cell are not there. And similarly for this next row and the next row. So these 12 experiments will do and try to estimate the ground truth that we have. Okay. All right, so with that, here is a method to answer this question. It's a method of synthetic intervention. And this is manuscript for the original method. What I'll do now is through pictures try to explain exactly what we are going to do in terms of data, and then applying method to fill in all of these cells. Okay, so let's look at the data available for this cell where I'm saying that I've got data available. How would it look like? Well, there are some patients of patient type 5. They have received treatment drug 3. For each patient, if I look at it, patient has 4 covariates pre-treatment, and then there are 5 measurements after the treatment started. And the patients who were compliers, for them, we have all the 5 measurements. Patients who dropped out, Some dropped out after 2 visits, some dropped out after 3 visits, and so on. Okay, so that's my data. If I look at data across this entire row, it would look like this. That is, I've got type 1, type 2, up to type 6. So type 1, type 2, type 6 patients. For all the patients, of course, I have preclinical covariates. But only for patient type 5 and 6, I've got information for compliers and dropouts, as indicated by these 2 checkboxes. But all of these values are missing, as indicated by here. And of course, these values are missing because people have dropped out. So at some level, what I really want to do is I want to fill in these missing values. And if I do that, then I'm sort of able to fill in these rows. Okay. Just to give another example, what I have here with the second row is I've got patient type 3 and type 4. Again, preclinical covariates information, treatment outcome for compliers and dropouts. These are missing because these people are dropped out. And again, as we know in this setting, people drop out because of the experiences they have, not just, at times they do drop out because of the logistical reasons. And that's a random dropout, and that's a good thing because that doesn't introduce bias, right? At times people might sort of drop out because they might get a treatment and they might feel that they're not getting better, maybe they're getting worse, and they just don't want to continue with the trial. Okay. And giving one more example, this first row corresponds to a matrix like this with all these values that you want to fill in. All right, so in a nutshell, now I have a problem. I have a problem with 3 different matrices that I want to fill in, okay? With the help of data available from those missing values in missing matrices and this additional matrix, which has information like this, with some values missing in the biased form. Okay, so now let's just look at the first order information in this data. And in particular, what I would do is that I would simply look at the matrices or aspects of matrices that are filled and try to understand what are, how does the spectrum look like? So if I look at a matrix of patients that receive drug 0 with rows corresponding to compliers only, corresponding to the 5 measurements I had. So I've got 28 times 5 matrix for drug 0. So if you remember, you might remember or not, that for the first drug 0, we had 28 compliers out of 36 or so people assigned to it. For this matrix, if you plot the spectrum, in fact, it looks like it is rank 1. That is, its largest singular value is pretty high compared to all the other singular values, which are effectively flat. Okay, and for this measurements, I'm not using covariates. Okay, the same thing holds for drug 1 as well. Again, 128 compliers, 5 columns. Drug 2, 124 compliers, 5 columns. Again, it's a rank 1 matrix. And the same thing with drug 3. All these matrices of compliers by measurements are effectively rank 1. What does it suggest? It suggests that in each of these matrices, each row can be written as some multiplier of any other row. Okay. Because all of them are effectively in the same subspace. Now the question is that, is this multiplier the same? Are the subspaces associated with these matrices are the same, or they're very different? That's one of the questions that next we would like to examine. So to do that, here is what we'll do, and we'll do some kind of an aggregation, because at the end of the day, entries in this cells of my eventual interest are average effect of this specific drug on this specific group of patients. So what I'll do is now I will do aggregation of these raw matrices, and we'll come back to those raw matrices in a second, but let me first do aggregation. So rows are my 6 patient types, which are these, and then columns are 5 clinical measurements. And for each drug, I have some data available. For drug 0, I've got data from all my compliers available here. For drug one, I have data available for the purpose of experimentation, only these two patient type, and hence only these two rows, and so on. Okay. What are the entries in each of the matrix? Well, a row corresponds to a patient type, let's say patient type 1, and here it's a first visit. Across all the patient type of 1 who received drug 1, during their first visit, what is their clinical measurement? Just average that. It's really average value of that. Okay, so that's my aggregated matrices from going from individual patient level matrices. And now what we'll do is we'll just stack these things together. And for computing the spectrum, I'm going to utilize all data just to understand the structure in the data itself. And what we find is that when we stack all of these things, again, for the whole stack matrix, 6 patient types and now 20, because 5 measurements per drug times 4, 20, this matrix is rank 1. So it's quite remarkable. That means that all of these matrices are actually sharing the same subspace. If you stack it other way, that is, don't stack it column-wise, stack it row-wise, same story. It's effectively a rank 1 matrix. Okay. So what this suggests is that the data is extremely low dimensional. And what we really need to learn is these multipliers. If we manage to learn these multipliers well, we might be able to extrapolate things really, really well. Okay. And that in a nutshell is what we do. And so now I'll describe just how one would go about doing it in a little bit more systematic manner, that specific aspect. I know I have maybe 4 minutes or so I think I should be able to conclude well in 4 minutes. So let's see that this is what we want to do. We want to find out what is the average effect for this cell, that is for patient type 1 under drug 3. Under my aggregated matrices, I've got these 4 matrices, drug 0, drug 1, drug 2, drug 3. I'll just stack them like a tensor. And now I want to fill in this row that would correspond to this. To do that, here's what I will do. You say, well, look, I know the row corresponding to patient type 1 under drug 0. I also know rows under for patient type 5 and 6 under drug 0. I know the data for patient type 5 and 6 under drug 3, but I don't know this. So what I'll try to do is that I will try to learn how I represent this as a linear function of these covariates effectively. Putting it other way, if it's rank 1, I'm trying to find multiplier that says, take any of the row here, multiply it, and represent it, you will get this row. Take exact same multiplier that you learned in this gray matrix and apply it to this green matrix, and then you will produce this answer. Formally, it would look something like this. Okay, you're just doing learning appropriate linear model, and then you are doing linear prediction. Okay, just another example of this. Let's say if you want to, in the second slice, you wanted to estimate this last row. You'll look at this in the gray one, precisely the ones that are the rows that are present in this one. Focus on them, try to learn this row as a linear combination of this. And then after you learn that, you do prediction. Okay, and that's in a nutshell is the method. One remark that I would, and you can sort of then after that, you can actually get the confidence interval and all that stuff. Underlying, you want to pre-process your data to remove the noise with principal component regression rather than doing just simple regression to allow for removing the noise. I won't delve into that. Just to give you summary result, how well does this work on the data? Well, with the exception of one setting where for drug 2 of a specific patient type I3, where number of data points were very, very few, effectively you're getting R square of estimation, which is above 0.8, okay? And sometimes even better. Going to individual level, and this is what I find really remarkable, that what we're doing here is that for a patient, we're going to completely withhold her or his post-clinical, post-intervention data. That post-intervention data is this blue trajectory. Now, using just that patient's pre-treatment covariates, we're going to use this method to predict this entire trajectory. And then, of course, the confidence interval. And this is what we get. And as you can see, across different patients, these things are very heterogeneous. And somehow method is able to get the right prediction. Okay. Going to the last part, which is, does it help understanding biases induced by dropout? So if you remember, drug 2 was the effective one where people were primarily dropping off because of logistical reason, like somebody moved and things like that. And as one would expect, that this was the distribution of change in score under compliers, and this was the change in score under our estimated dropouts. And this pretty much coincides. On the other hand, there's another treatment for which lots of patients dropped out, and the dropout are the ones that were doing poor performance. So really, if we had included this, the drug 1's performance would look much worse compared to what it was based on compliers. So in this case, one could actually sort of see that dropouts have significant bias correction that's happening there. Anyways, I think I'm at the end of my time. It's a good place for me to stop and ask for questions.
Thank you so much, Devavrat. So please, everyone, just put in your questions into the Q&A. And maybe, Devavrat, I can start off with one on, your subtype groups might be very different sized. Where do you see that showing up in your confidence intervals, I'm assuming? And also when you showed the trajectories, was this for individuals from the same subtype that can have so different trajectories?
Yes. Okay. So maybe 3 questions, right? Or 2 questions, sorry. So one is about confidence intervals. So you're absolutely right. The confidence intervals would change depending on the number of donors you might have, and the amount of data you have and all that, and standard deviation, how it would look like. So that's perfect. Coming to the second part, yes, so those trajectories were individual level. So imagine that I have, let's say I went through treatment 1.
Yeah.
And that means that sort of I know my entire trajectory. Of course, I don't know my trajectory under treatment 2, 3, or 4. So that's fine. Sure. I'm just going to withhold myself. But for treatment, patients who have gone through treatment work, I'm going to sort of, let's say, utilize a small number of patients. For that experiment, we utilize 50 patients to build the synthetic model based on the covariate information. So effectively, we thought of covariate information as observation as well to build the synthetic intervention model, and then sort of simply did the linear prediction.
I'm just wondering if I would take a random person that has the same subtype, because you left them in your training set, a random person of the same subtype with the same drug, how would that curve look?
Beautiful. So that was that R square. Okay. That is, indeed, you would look sort of very different. Putting it other way, let's say if I looked at the distribution generated by 50 random patient of a given type, okay, and I will plot the distribution, and then I will, for those other 50 let's say other 50 patients that I want to actually use synthetic intervention, and plot their distribution, and look at then I have a true data. If I look at the, let's say, the Jensen-Shannon entropy measurement, the RCT distributional estimate would be like something like 0.6, 0.7, and you have some statistics there. On the other end, this one would look like 0.05, 0.06.
Okay. Wow.
So there is a pretty serious heterogeneity in the data.
Super exciting.
Yeah.
And then maybe one question which will also lead over to Regina's talk is, in the end, what will be exciting is if you could also say something about a new drug. So here you always need some entries in every row and every column.
Yes.
How would you go about that?
It's a great question. I think maybe also connects to some of your things that you are speaking and things you are doing beyond. And here, if we learn these latent factors associated with interventions, and somehow if the interventions had features, biological features or whatever, that one could connect them to these latent factors, then maybe one could potentially expand or extrapolate to interventions that one has not seen before.
Yeah, that could be, for example, molecular structures that we might hear next by Regina to actually relate them. I think that would be really exciting. Great. Thank you very much, Devavrat. It was a real pleasure. And now I hope, Regina, could you say if you're here?
I'm here.
Yay!
Hello.
Awesome.
Hello.
So then, in that case, I will introduce you. It's a real pleasure to introduce Regina Barzilay. She's the School of Engineering Distinguished Professor for AI and Health in the Department of Electrical Engineering and Computer Science at MIT. She also leads the Jamil Clinic, which is an MIT Center for Machine Learning in Health. And her research has really revolutionized our thinking in many different biomedical areas. And most recently, and I think that's what she will be talking about here, in the development of machine learning algorithms for modeling molecules for applications to drug discovery and clinical AI. And with this, I'll turn over to you, Regina.
Share. Uh,
Awesome, yeah. Sharing the
okay,
screen.
it's sharing something. Okay. Can
And
you see my
perfect.
slides?
We see
Yeah.
everything. Yep.
Yeah. First of all, thank you very much, Caroline, for such an introduction. Truly honored to be here. And I think that, the talk that I'm going to present is really complementary to both Caroline's and Devavrat's talk. And it's going to focus on the molecular models because, once we identify the targets that we're interested in, as Caroline was presenting, we really want to be able to drug them accordingly. And in this talk, I will primarily focus on small molecules, but we do also research on biologics, but kind of to stay coherent within 25 minutes, I will talk about small molecules. But at the same time, kind of understanding what these molecules do is really important to be able to generalize. And I think I would be able to answer the question that Caroline posed to Devavrat. In other words, how we can generalize in the molecular space and not treat different drugs just like kind of a symbol, because there is clear connection between the structures. Okay, so since I'm talking about this whole space of, molecular modeling, I would say it's very broad space, and I would tell you what I'm going to be doing there, but kind of the MIT group actually looks at all these different aspects. So maybe the most advanced area, which is currently used already in pharmaceutical industry and chemical industry, it's a property prediction. So this is a capacity to take molecule and in silico predict different properties depending on your training set. It can be some physical chemical property like solubility, or it can be some activity on the target or toxicity. So the second big area in this space, in molecular space, is actually predicting reaction and finding the most effective way to generate molecules of interest. There is an area which is really in the intersection of, my research and Caroline's research, and we have actually a joint paper in this space of how do you jointly model the chemical perturbation and, biological properties. This is an area that is still, developing, but I would expect there will be a lot of interesting advancements in the next few years. And the final area, but definitely not, maybe one of the most important one, is actually how do you de novo generate the drugs? How do you, to answer the questions that was previously posed, how can you generate molecule that, for instance, interacts productively with multiple targets? So I will be primarily speaking about the property prediction one and learning molecular representation, but I will also allude about some biological properties and talk about a bit about generation. So I want to say that, this talk is kind of reflects my personal progression in this space. When we started working on it, 5 or 6 years ago, this field is really very junior in terms of kind of machine learning field. For me, there was a lot of learning how I start thinking about it and where we are now. And I want to kind of share with you some of these observations. So I want to start, with something positive, and maybe the most positive example of what we produced so far, we utilize these models to identify a very potent antibiotic molecule, which is described in the Cell paper. It even has its own Wikipedia page. And this particular molecule has a novel mechanism of action. And it's shown to be very effective against multiple pathogens which are drug resistant. So, and the way, like, to bring it back to the computer science side, it was actually a very simple, in some ways, computer science experiment. So Jonathan Stokes, who was a postdoc at MIT and now he's a professor in McMaster University, scanned 2,500 molecules against E. coli. So you can see the result of this screen below, which kind of indicates for different molecules how active they were against E. coli. And then you can actually train a model given this data where on one hand you have molecule, on another hand you have this measurement of antibacterial activity. So now you could actually take this model, screen humongous chemical space, and identify potent molecules. That's what was done there. But one question that especially more biologically inclined people in the audience may be thinking about is that even if you're looking at antibacterial activity, we know quite a bit. There are very complex processes that can actually enable a molecule to be antibacterial. So you can say how we can take all this biological knowledge, kind of remove it whatsoever, and just give to the model the structure of the molecule and the outcome. So what I will do, I would start by showing the models which are sort of really biologically naive. And as we continue in this talk, I will show to you in which scenarios this kind of thinking is really insufficient. So in some ways, at the beginning of the talk, I'm not going to be really looking at the interaction between the targets and molecules. I am going to do a variety of simplification. So for instance, if I'm thinking about how this molecule and the target interact, I would say, just collect for me the data, what this molecule does for a single target, or maybe collect me the data, what molecule does in some phenotypical screen. So my input would be not the full interaction. My input would be the molecule and the number. And then I'm going to do even bigger simplifying assumption, even though we know that clearly, the 3D geometry really plays a significant role in activity. I'm going to say, forget about it. We're going to model it as 2D graphs. So the input of the model will be a 2D graph and some number that, that we want to capture, that in this case captures antibacterial activity. Now, the idea of how to land the mapping from 2D graph to the number is quite old. And if you look at a more traditional kind of medicinal chemistry approach, the way they've done it, like for decades before deep learning came into a play, was the following. You take molecule and you summarize it as a fingerprint. So for the computer scientists in the audience, what it means that you are pretty much summarizing molecule as a vector of different substructures. And now you have this vector and you can run your favorite classifier and predict the property. The problem, as chemists themselves discover, that many times when you're looking at some interesting properties, you don't really know what are these features that are really important. And that's where the deep learning came in. And people really start thinking, maybe instead of kind of starting with the handcrafted vectors, we can lend them directly, these vectors, from the molecule. And it worked pretty well, of course, for computer vision, for natural language processing, and indeed it works pretty well for molecules. So what you would see here is that if you completed this process of abstraction, you're going to see that molecules that have similar property lives in kind of close path. Their vectors in high dimensional space would be close to each other, and the ones which are very different would live in different parts of the space. And of course, you would build this representation for a specific property. So if the same molecule for toxicity and for, let's say, solubility, obviously would have different representation. And though I didn't really, I'm not going to talk a lot about generation, but of course you can imagine that once we created this latent space in which molecules live, you can, for instance, imagine to yourself doing lead optimization. You can imagine I'm starting in some particular place, I'm going to now optimize it with my property predictor and generate a new molecule. And coming back to the question that was asked earlier about doing it multi-target, absolutely you can. If you have predictors for binding to multiple targets, you can actually generate a molecule which binds to all of them or selectively binds and so on. And I just very briefly want to tell you how this representation is done. What I would show you sort of in some ways is a caricature, because there is like, even for this class of graph convolution model, there are more complex models, but it kind of captures the intuition of what's going on, so it doesn't look miraculous how it came about. So you can imagine what you want to do. Again, you want to start with a 2D graph and you want to translate it into a vector. So you start by initializing every atom with a corresponding vector. And here you can ask your expert, which features of the atom they want to use. Here on the slide, I've shown you 4 features. In reality, I think you could have much bigger set of features. But when you've done that, every molecule only knows about itself. It doesn't know about the neighbors, which we know it's really crucial. So in this case, what you're going to do message passing. So every molecule would have its own vector. The neighbors have the vector. You can actually learn how to combine them together to get a new representation for that atom. And again, this combination would be guided by the desired property prediction accuracy. So within one hot, you're going to look at your immediate neighborhood. But since everybody get updated, the information from a bigger neighborhoods to hop neighborhoods are going to start trickle in, and eventually every molecule would not only know about itself, but it also would know about kind of surrounding friends. And at the end, what we can do, like this is the simplest way to do combination. You can just take every vector for every molecule and sum them up. And this what would be used to predict the property. And I want to preempt the question, which always comes. Is it, can you use transformer? Can you use something else? Absolutely. There are lots of different paradigms, but the basic idea is very similar. That's what these models try to capture. There are a lot of kind of different architectures in this space. I'm showing you just one publication that we've done that is very commonly used today as a baseline. It works, pretty reasonably, but I'm pretty sure that as I'm speaking, there are already, better models out there. But we very extensively tested it. And what we've seen, again, that it works pretty reasonably if you have a large training set. But of course, performance goes down when you're learning representation, when your training set is very small. There are very few positive examples. So there is no surprise here. And this is a challenge for all different types of models out there, especially those with the more complex architectures. And now I want to go back to this picture that I show you in the beginning of my presentation. This is the mechanism of action. So for instance, for the antibacterial, there is a lot of information of, how the existing FDA-approved drugs are achieving their antibacterial effect, with which targets they operate and so on. So the question that actually our biology collaborator, asked us, can you predict, given a new molecule, what will be the biological mechanism of action? And this was not kind of a trivial question, because, once we identify this halicin, this molecule with the desired property, he spent a good few months trying to elucidate the mechanism of action. And apparently he said that even though he spent a few months, he was lucky that he eventually discovered it. And the answer to it, yes, to some extent. So we know that there are many different sources that, again, for all the FDA-approved drugs can tell you exactly what are the targets, what biological processes are involved. So the same way as until now I demonstrated, you can use this representation to predict, activity. You can also give answers to all these questions, what target is involved and so on. Now, my personal view on this, and you can do it with very reasonable accuracy. My personal view on it is And I'm not sure it's very productive direction for various reasons. We know that whenever you look at these repositories which record this information, that even for FDA approved drugs, the information is extremely incomplete. And it's not only incomplete kind of randomly, there is a selection bias of what targets got tested. Uh, so in some ways, whenever you're training the model, you're introducing human bias, but on the other hand, you don't really revealing a lot of new stuff. And my personal preference, that's how, I'm hoping to do more work in this area, really think about the mechanism of action through the imaging, where you can see what interaction happens, and you're not limited to a summary when you say it was this target but not that target, where you can see the full picture of what's going on. And maybe you cannot really exactly kind of explicitly state which targets are involved, but you can capture so much, and you can identify which molecules have similar mechanism of actions. And it's particularly relevant when we're thinking about, chemotherapy drugs. We know that the vast majority of patients are treated today by the drugs that were developed, decades ago, which are very dirty drugs in some way because they operate with multiple targets, and for many of them we don't fully understand the mechanism of action. So this kind of view, using imaging and identifying molecules with similar mechanism of action, I think would be really impactful to identify these new potentially interesting classes of drugs. But let me go back, as I promised to you, that I want to go beyond just looking at the molecule as 2D structures. And I want to get closer to this picture where we kind of understand what the small molecule does to the target. And until the pandemic started, I was very squarely in small molecules world. Like chemistry was the main thing. I never saw that there is much to do. It's not that there is not much to do, but I wasn't really thinking about the biological targets. We were really playing in small molecule game. But the reason we had to change our ways and kind of to see the light came because of the request from Ann Katz, which is one of the institutes of NIH, when they asked us to find the synergetic drugs for remdesivir, for one of the antivirals that was popular at the time against COVID. And we know that combinations are very effective against antivirals, like for HIV. So the question is how you can identify these antivirals. And this work was done by our prior student, Wen Gong Jin, who is now a postdoc working with Caroline at Broad. The key challenge here, why we couldn't directly apply the same architecture, is because the amount of data we had. While we had plenty of data for individual compounds, like we could take a molecule and we can predict its antiviral property, we had only 200 points for combination. So clearly you cannot directly learn, like proper deep learning model on 200 points directly. And the particular properties that, again, we are trying to optimize its synergy. And, here you can see the formula. It's called Bliss formula. You want to identify a combination where the sum is bigger than its parts in colloquial terms. And when we were trying to think what we can do, we kind of start noticing that at the time there were already a lot of papers published about relevant targets for COVID antivirals. There were quite a number of nice papers where, of course, none of them claimed that we understand all the targets, but at least there was like a clear empirical evidence about a subset of these targets that play a role. And I should say that there are resources like Campbell where you have information about a single compound and its activation of the target. So let me just summarize what we have. We have for single molecule sufficient amount of data to learn its antiviral property. And we also have data about, for all of these interesting targets, which molecule activate them or not. And this has actually brought us to this new view of trying to understand whether the combination is synergetic when we are reasoning about targets, because we know that synergy is achieved when you are looking at multiple targets which are involved in this process. So here I'm showing you the picture. It's a different graphics, but the same idea. What you already seen with this graph convolutional model, you start with a molecule, you do this message passing, you summarize it into a vector and predict the antiviral property. So what we will do now, we would actually incorporate our information about targets. So what we will do, we will map our molecule to learn which targets it activates, and then from these targets you're going to predict the antiviral activity. So now target is active part of the prediction. So if you're looking now on my slide, you can see that the representation itself has two parts. One part are the targets themselves, showing as a triangle, but there is also another part of the vector which doesn't directly correspond to targets. And the way I'm thinking about it is, the kind of the structural summary of the molecule, what we had before. The reason we have it, that you cannot explain everything with target, because we know again that our information about the targets is incomplete. And this way you're both kind of in your representation keeping biological target properties and also the chemical properties. So it's kind of a biochemical representation. And then what the model does, you kind of train for each one of these compounds, you learn this target structural representation. And then when you have representation, which is the bulk, the hardship of our learning, then you can concatenate it together and learn the bliss, the synergy score. And again, because you already learned the representation, which is very small, you can do it successfully even on 200 combinations. And I should say that it worked quite successfully. Overall, the accuracy was 82, which is not bad. And we identified for remdesivir several combinations which researchers and NCATs tested empirically and demonstrated that the combination were highly synergetic. As you can see, in one case, the combination killed 100% of the virus. And what I want to say, to me, it was really an eye-opening, discovery that the adding these targets, understanding at least in a minimal way what's going on, really improves the accuracy. And here I'm putting a reference to PNIS paper where we do these experiments that show that the more targets, the more accurate target information you are including, the better your results. And I know, again, that there are people, maybe Caroline and others, that are kind of thinking, wow, you are introducing your targets in such a stupid way. They're just one hot encoding of the targets. And that's exactly where we are going next. That even with a simplistic encoding of the target, you're already showing some interesting result. The question is how far you can push it if you're actually looking not on the structure of the molecules that we kind of generalize it, but also if you're really looking at the full understanding of the structure of the protein. And I want to mention a paper that was very puzzling to me. And it was during the time when I kind of made a foray into this whole target business. And this is a paper that comes from colleagues from Boston that kind of look at these networks which connect molecules and targets to make prediction about drug reuse. And the idea here is simple. You kind of say, okay, I have different drugs. I know which targets they activate, again, for FDA-approved drugs. I also know there are protein-protein network. I know the relation between the proteins, and I know the relation between the proteins and disease. So now I can kind of use, any types of kind of graph reasoning approaches to predict the interaction between drug to some really new targets and potentially identify new treatment for diseases. Now, when personally I look at this paper, which reported very high numbers, I was a bit puzzled for two reasons. First of all, this representation doesn't even use a representation of molecules. It's all symbols. It's all one hot, molecules, proteins, everything. And second of all, as we've said earlier, that these graphs are extremely incomplete, and they're incomplete in a biased way because a lot of things don't get tested. So I was really wondering, how come it reports such a good results? And here I asked one of my PhD students, Rachel Wu, to kind of reproduce these results. And we emulated them on this depth map data where you have 4,500 drugs and you have cell lines which kind of you can treat as a diseases. And what we've discovered, I would just, since I'm close to the end of my talk, I just want to say what we've discovered, that it's very interesting that it really depends how do you divide your training and testing data. So the results that were reported in the paper considered the easiest kind of breaking, where you have the set of targets and molecules and the diseases is the same for training and testing. You can just take new combinations. So it's kind of random split on the triples. But if you start being kind of make it more realistic, and you say, I really want to generalize to new targets. So I want to generalize to new molecules. In this case, the performance of this model fails dramatically. So you can see you're going from 94, it's a very last line, to 66. So and this was again the lesson for me looking at it. First of all, you need to make sure that you are generalizing correctly, but also that you are creating training and test speed, which is really realistic, but also it's really important to look at the underlying structure. And I just want, at the end of my talk, say that, our current work in the last in the last year actually focused on truly going, not only saying that we have a 2D molecule and some target, really fully understanding the interaction between the molecules and the targets. And we've demonstrated that we can do many challenging tasks like docking, like binding, much faster now, like sometimes up to 500 times faster with a comparable quality. Here I'm just showing the work of Octavian Ganea, who is the postdoc that Tommy and I are co-advising. You can see some of these papers, and it's a really exciting time. And I just want to close by showing you, a good example. So each one of these papers reports a big evaluation, but Patrick Walters is one of the really leading scientists in the world of molecular simulation. He is a VP of Relay Therapeutics. So when Octavian shared with him the paper, he kind of said, I want to see whether it can handle really tricky scenarios, and all the other models, including Schrodinger's model, which is a commercial model, fail. And this you can see two of the Novartis drug, and it shows where they should be docking. And we try, so Patrick was very correct, the model fails, like Schrodinger model, and the other fails on it. And our model was actually able to correctly identify the placing. And the reason I'm showing this example, because kind of Patrick saw that it's super challenging. So just to conclude, I want to say that, my main learning myself was that in order to be really meaningful in terms of therapeutic development, it's important to test generalization correctly, and doing random split that we do in many other areas of machine learning may not be the right thing. You need to make it even harder. The second one, I think that we can generalize better and handle the scenario where the data is not available by really producing much richer representations that look at the geometry of molecule target interaction. And finally, we need to think, all of us, harder how to think more about more complex mechanisms of action, how to inform our models, and what we as machine learning people can say new to people in biology about the mechanism of actions, which is really a challenging task. So thank you very much. Let me
Thank you so much, Regina, for a really wonderful talk. Again, please put questions into the chat if you have any. And I want to actually just, if you have 5 minutes, Regina, to talk briefly about what you ended with on, mechanisms of actions, and in particular, like the end of your talk and connecting that to the beginning, where you're optimizing for particular properties, which is a very small dimensional vector usually. But then if you're, when you're showing that, of course, a drug can have very different effects in different contexts, say in these different cell lines. So, and that would relate to mechanisms of action. I think if you would know the mechanisms of actions, then you should be able to generalize across different cell lines. So how is it possible that one could get such good results by just optimizing a particular property without taking that into account? So without taking into account that you need to understand then afterwards that a drug can have very different effects in very different contexts?
Excellent question, Caroline. So let me just explain to you again. It's just my I don't have empirical results to support it, but let's say you have a particular target in mind, and you have certain geometry that will get into the right binding pocket for this target. So what it means on the molecular view, that there will be certain, substructures, molecular substructures that will fit there. So in a sense, you don't really understand, where it's trying to fit, but you would relent the correlation that certain substructures are likely to fit there without understanding what is there. So you can actually do it for one property at a time, but you really cannot generalize to something new. This is a problem, that you're making this kind of, it's not causation, it's more correlation, that these substructures happen to fit here. And if you have a very large data set that illustrates multiple examples of this kind of fragments that are likely to fit in there, you're going to learn it. But at the moment that you don't have enough of it, or you're sampling from your training set comes from a very different, from a very large set where there is a wide range of possible fragments, you may not be able to do it well. And following your question, I just want to kind of say how, to me, it's like a mode of failure of this model to some extent. As we know, in natural language processing and in computer vision, pre-training plays humongous role. And like, for instance, the closest you can say to pre-training in this case would be to take a graph and like mask some parts of it and try to predict it back and the other things. So these models, for the longest time, they didn't deliver anything. Now they predict, this pre-training gives you really a few points, but it really doesn't do the same kind of miracles we see in NLP, where nobody would even consider doing the stuff from scratch. And this is my point, that we can, and there is like not a lack of people who are really trying to do more and more graph pre-training. But my point is that unless you incorporate correct biology into it, and you know what you are doing here, then we're just going to do more graph pre-training algorithms, which would not help to solve molecular problems.
Thank you, Regina. And there is also another question, which is, related to that. Often clinical trials fail in the end because we see a large number of off-target bindings. And as the effect of off-target binding increases, toxicity increases. So how do you think about, directly building in, of course, this as an objective function of thinking about toxicity?
Yeah, so I will start first by saying that this is really a great application. And then, Caroline, I would let you continue, because that part of your research is quite relevant to it. But let me just start and tell you what we can do on our end. We already know that if you have a good toxicity data, you can train the models to predict toxicities. That's what we did with halicin, like generic toxicity or some particular genotoxicity or some particular type of toxicity. And you can kind of predictively say you shouldn't be even going into that space, so you can optimize it. You can also have the models for particular target of interest. Let's say you look at all the rest of the proteins and you see, is it likely in silico? Is it likely that this molecule is going to bind it and create the side effect? And as far as I know, in many pharmaceutical companies, they're using ChemProp and equivalent model and the binding model to achieve that. I also believe that as we become much more refined in our kind of what we are capturing, because we know, for instance, where we're thinking molecule goes into the cell, but where does it go into the cell? Is it going to nuclei? And other important questions. We have ability, still limited, but we're getting there, to predict and to understand what's going on. And again, in silico, eliminate, bad actors before they go into a trial. But I want to actually let Caroline to say about some really interesting research that she's doing, which relates to it. As we know that one of the biggest challenging in translation is the fact that, we do it like on animal models, but then surprises come when we move from animal to humans. And this brings me to really exciting research that Caroline is doing, is, what you can say when you are moving across different types of cells. Because right now it's very empirical question. So Caroline, I'm sorry to put you on the spotlight, but I think it's really fascinating to hear that.
Thank you, Regina. Yes, and actually it relates very much to what Devavrat has been talking about. And so maybe we can also close with that on, in fact, if we're looking at the cell, what he had as disease or disease subtypes as being, different kinds of, say, cell lines or cell types, or in even different types of organisms. And then on the other hand, where you have different types of drugs, and you're trying to actually go across different disease contexts or across even different organisms, then you get to these really combinatorial questions of, you will never be able to do all of these experiments of all of the drugs in all of the different contexts that you actually care about. And so synthetic interventions is one approach to try to get at that. And so I think we had a nice cycle here in this presentation of trying to think through targets in the genetic setting first, and how, what the effect is of targeting different types of genes and how to optimize over that. Then thinking through, how to explore this huge space of combinations of, say, different interventions and different contexts, and how to move between them. And now with Regina's talks on actually trying to build these molecules and how to explore this space of molecules that can really then actually target the right targets that we may want to target with the correct properties. So with this, I want to thank Regina and Devavrat again, and everyone here for listening and the great questions. And with this, we'll end the session here. Bye, everyone.
Bye.